This afternoon's talk will be given by Peter Daszak, but before I introduce him, I first want to acknowledge President Dennison for his support of the IGER program that we have on campus, the Montana Ecology and Infectious Disease Program, in the form, in this case, of allowing us to nominate a speaker in the area of disease ecology each year for the presidential lecture series. And in this case, this is something that the trainees in the IGER program put their heads together to decide who they want and basically make arrangements, etc. So I wanted to acknowledge the president. I want to also mention that there is another talk tonight, the sort of town and gown talk at 8 o'clock in the University Theater by Dr. Daszak. And I guess with that, by way of introduction, Peter comes here from the Consortium for Conservation Medicine. He actually did his schoolwork in London, well, in the UK anyway, getting his bachelor's degree in zoology, and then moving on and doing his PhD in parasitology at East London University, studying coccidia, after which he came here and spent some time at the CDC during the Nipah virus outbreak, and then University of Georgia? Yeah. Georgia? Studying Chytridiomycosis, the fungal parasite that has been identified as responsible for the amphibian decline in the US. So Peter was deeply involved in that research. He now is the executive director of the Consortium for Conservation Medicine in New York, and also has adjunct appointments at Columbia and at Tufts. And as you can see, today he's going to be talking about why emerging diseases happen and what can we do to stop them. Peter?
Thanks very much. I'll be off then. Oh no. It's a great honour to be here, and I appreciate the being the student's choice. That means a lot to me. It's very nice here. And as you know, I've lived in different places in the US, so you have a choice of 3 accents. I can do this in a New York accent, an English accent, or a Georgia accent. So, okay, English it is, right. So basically what I want to talk about is work that we've been doing for the past decade now on trying to mix different disciplines together around the field of emerging diseases and get to a position where we can deal with this significant problem. And I think it really is a significant problem for 2 reasons. First of all, these are the really big diseases that are hitting us now in our population. Diseases like HIV/AIDS, where 40 million people can be infected and die of what is essentially a chimpanzee virus. So something is going on there. And also look at the economic cost of some of these pathogens. If you look at the virus I'm going to talk about in a bit, Nipah virus, $350 to $400 million in one outbreak in one country. But when you look at some outbreaks that hit livestock or multinational industries like SARS, you end up in the billions of dollars, $50 to $100 billion in one single outbreak. And the projections of what the economic cost of a single outbreak of pandemic avian influenza, if avian influenza went human-to-human transmission and started to move around the world, is somewhere between $350 and $800 billion. So not only are we talking about lives lost, we're also talking about significant hindrance to our economic growth and our business on this planet. So it's something that we should do something about. Why are we seeing this high impact and why are we seeing this growth in diseases that are appearing seemingly out of nowhere? Well, one of the reasons is, of course, that we travel around the planet at an unprecedented rate. Thanks. So we have here, this is a map of 1 year's volume of air travel, human air travel around the planet. And as you can see, we do the most travel here, the richer countries around the world. But the network is incredible. It reaches into all the sites where diseases emerge. And once a pathogen gets into that system, it's very easy for it to move around the planet. But knowing that these diseases are driven by things that we do, like global air travel or trade or changes in demography, means that there's a way we can start to understand them better. Now, an emerging disease is a disease that's on the rise. It's a disease that's moving from one host to another or from one geographic region to another. And that is driven by essentially two factors: human demography, changes in our structure, our social structure or behaviour. And a good example of that is HIV/AIDS, which spread through global travel, air travel across the world, which was originally driven by bushmeat hunting and changes in demography in Africa. And it's also driven by, they're also driven by environmental change. So a great example of that are diseases like influenza, for instance, which, when you look at the pattern of poultry production in China and other parts of Southeast Asia, a significant increase over the past few decades, and this intertwining of pig production and poultry production that allows the virus to exchange genetic elements and develop new strains. So if we know what drives these diseases, my hypothesis, we can predict them, we can predict their emergence. And what we need to do is look at this in really three different levels. The first level, and the easiest to predict, is actually the rarest event. It's diseases like HIV and SARS which go pandemic, become global. And these are diseases that get into the human population, travel through either our own travel or our trade patterns around the world. Like West Nile virus, for instance, probably came over to the US either in mosquitoes on an aeroplane or in birds brought over here for trade. And that's relatively easy to predict. An earlier level of emergencies gets to be a lot harder to predict. I'm going to talk a little bit about one virus, Nipah virus, which is in the early stage of becoming a pandemic. And this is represented by spillover from wildlife into people and these little green oscillations. Small localized outbreaks that occasionally become big events, as happened recently with Ebola, Marburg virus, that went into a more urban area and became a larger outbreak. If those viruses get the potential to evolve and become adapted to humans and start to enter this human-to-human transmission chain, then they can move to this pandemic state. But that's quite hard to predict, and I'll show you why in a bit. And I think that what we really need to get to is this pre-emergence stage where, and specifically for these diseases like HIV and SARS that come from wildlife originally, they're the most common group. What we're going to see when we change the environment, we're going to see pathogens in one wildlife population spilling over into another and then making this pre-emergence stage before they spill over into people. So where we see outbreaks in wildlife, we're seeing a signal of something that could lead to an outbreak in people. So getting to the level of understanding what this means and predicting that should lead us to understanding how to predict, ultimately, this pathway. At least that's the theory. Now, let's start with the pandemic thing. I want to show 2 or 3 examples of how other groups, and our group in particular, have been trying to predict these different stages of emergence. So this is a really nice paper in PNAS by Huff-Nagel et al. That shows if you just simply measure those global patterns of air travel, you can very easily back predict the number of cases per country for SARS during the outbreak of SARS. Because SARS was a human-to-human transmitted pathogen once it became an epidemic. And we know where it originated, and we know the travel patterns from China, Hong Kong, into other regions. So you can pretty easily predict it, and actually incredibly accurately in this paper. So the number of cases in Hong Kong was 1,718. In the simulation, the minimum was 1,400-ish, the maximum 2,700. And as you go down the list, incredibly accurate predictive capacity. So once you know that, you can begin to predict what would happen if the next outbreak emerged here or there, and then do something to stop it. But that's relatively easy because this virus moves from just humans to humans. There's no involvement of other animals or other pathways. It's a straightforward single host issue. What about a disease like West Nile virus where you've got birds and mosquitoes and then people? That becomes much more complex. But I want to show a couple of things we've been doing. First of all, predicting where West Nile virus is going to go next and how it's going to get there, and then trying to predict the impact in a specific region of West Nile virus every year. So West Nile virus emerged, it's an old world virus. It circulates between Europe and Africa in birds essentially. Humans are a dead-end host. We don't transmit the disease to other people. And we're sort of spillover. We're a sort of dead end for the virus, a false result. Now, in 1999, the virus suddenly appeared in New York. Now, that's no chance event. This virus has been circulating in Europe and Africa for millions of years, probably, or certainly for a few million years. Why did it suddenly appear in New York in 1999? So that's the key to understanding how it's then going to spread from New York. So we started to think about this and how easy would it be to predict, not necessarily where it's going next, but let's look at a virus that's now spread across the US and now is down into Central and Southern America. What about Hawaii? Now, West Nile hits birds, it kills them. It also kills people, so it's going to be a significant impact if it gets to Hawaii on tourism, because it's not good to go to a beautiful island where you can catch a disease that kills you, and also on birds. There are some birds there that may be susceptible. So this seemed like a valid idea to look at this. And actually this was work that a postdoc of mine, Mark Kilpatrick, really wanted to do as a sort of side project. And then I thought, well, this is a great project, because we actually ended up going to Hawaii, talking to, so we got a trip to Hawaii. We met with the government and they talked about enacting some measures to stop it getting over there. Then he said, what about Galapagos? What about Darwin's finches? Critical, the tourists go there to look at Darwin's finches. It's the third biggest earner of foreign GDP for Ecuador. This has some potential policy implications. Plus we get a trip to Galapagos. That was great. So we did that as well. Then he wanted to work on Barbados. I said, wait a minute, hold it right there. And he did actually do an analysis for Barbados, but we didn't get the trip. So this is what we found. If you really want to predict how this virus is going to get to Hawaii, and by doing that then you can prevent it, you need to look at 5 different pathways. Infected people carrying the virus, mosquitoes blown across on the wind, human-transported mosquitoes either in boats or airplanes, human-transported birds essentially taken over as pets or livestock, and migratory birds. Now these things seem very far-fetched, but on the flight over to Hawaii to discuss the results, there were mosquitoes on my plane and a parrot. So it just shows you these things do happen. I kid you not. I thought, oh yeah, that's one of our statistics right there. So how do you do it? Well, it's very simple. You need to know that for any of the pathways, the number of infectious individuals likely to get to Hawaii via that pathway per year, multiplied by how long they're infectious for, because that gives you a measure of the real risk. If an animal is infected but it only lasts a day, it clears the virus, that's a lower risk than an animal that's infectious for 10 days. So for mosquitoes, you also need to know the number of aeroplanes and boats. You need to know how many mosquitoes on average on a plane or a boat. And you need to know the prevalence in the place, the number infected in the place where the boat originates. Now for birds, it gets a bit more complicated because West Nile virus can infect over 100 US bird species. Some of those birds are very competent at transmitting the virus. When they get infected, they produce a lot of virus in the bloodstream, and therefore mosquitoes that bite them are very easily infected. So that's called vector competence, reservoir competence. So you have to have a figure in this equation for the range of competence for those different hosts. So it becomes a bit more complex, but it's essentially fairly straightforward if you've got the data. Now the good news is people aren't a problem. And I studied 2 subspecies of humans, the American tourist here, Homo migrans variation americanus, very easily spotted. And this is the British tourist, Homo migrans vulgaris, a common species in Hawaii too. Note the very severe sunburn. But the good news is, when we get infected with West Nile, the maximum viremia in our bloodstream isn't enough to infect the mosquitoes that live in Hawaii, or for that matter, in the US. So tourists are an almost negligible risk. In fact, the biggest risk, and this is a log scale, so that's a really significant risk factor. That's 2 to 3 orders of magnitude higher than any other risk. And this is for Galapagos, Hawaii, and Barbados, is mosquitoes landing on the island via airplanes. And if you work out the math, for Hawaii, it's between 6 and 60 infected mosquitoes per year. Now, it doesn't sound like a lot, but that's forever. That's for the rest of humanity, because West Nile virus is pretty much here to stay. It's not going to disappear. It's an endemic disease now in the US. So it also gives you a way to target control. And when we talked to the Hawaiian state government and the USDA, the conclusion is there is a way to stop this. Now, the average plane carries something like 1.2 mosquitoes. Now, 86% or something like that of those mosquitoes are found in the cargo hold, not in the cabin. So you don't need to spray the cabins. You just need to put residual insecticide in the cargo hold, and that should really reduce this risk. So we had a great result that was policy relevant and took just a few weeks to do. Not a lot of work and not a lot of funding required to do it. Oh, here are the data. So 1.55 live mosquitoes per plane. In fact, if you've ever been on a plane and they come down and spray the cabin, there's a really nice study by some of the New Zealanders that showed no significant difference between sprayed and unsprayed cabins, so it's pretty ineffective. You've really got to get to residual insecticide on the surfaces to make any difference. Now, at the time we were doing this analysis, avian influenza was becoming a hot topic, and we were saying, well, can we extend this analysis to avian influenza? The big issue with avian influenza for us here is, how's it going to get into the US? Well, can you extend this approach and predict the most likely pathway for it to get in the US? So 3 potential pathways. One is through the poultry trade, with live birds being moved around from country to stock farms and for slaughter, or via migratory birds again, or via birds in the pet trade, legal or illegal. To deal with it, we just needed data. So we needed data on live poultry, wild bird trade, and migratory birds. And there are data. You need to produce a risk measure, again, just like the West Nile virus analysis, the number of infectious bird days, the number of birds multiplied by the percentage infected and by how long they shed virus for. So we've got really good data on migratory birds, and these data from Royal Society for Protection of Birds and from Smithsonian. The numbers of birds in summer breeding grounds that migrate over an outbreak of H5N1 into a country that then gets infected at the exact time of year that that happened. And that hadn't been done before, to really go into that detail and look at the timing of the spread. And then we also had country-to-country trade in poultry from FAO. So what we end up with is, this is a map of what's happened so far. So every circle is a separate spreading event of avian influenza. And if it's blue, it means the most likely pathway is via migratory birds. If it's orange, the most likely pathway is via poultry trade. So what you've essentially got is a virus that emerged in Southeast Asia, in China actually, and then was spread around Southeast Asia through poultry trade. But then once it reached a few different countries, it started to become available to this global network of migratory pathways. And of course, poultry are kept outside quite often, next to water, ducks land, pick up the virus and take it on. And then you get this rapid diffusion of the virus across Europe by essentially the bird migration. Now this is important to us as a conservation group. People were starting to target birds in Europe and say, what are we going to do to eradicate waterfowl from these wetlands to protect us against H5N1? Well, that seems to be true. You know, birds do carry the virus, and it is an issue. But of course, if we hadn't had this huge expansion of poultry trade in Asia, the virus wouldn't have emerged. But the real issue here is, how's it going to get into the US? Now, the US government at the time had a blanket ban on poultry trade with any country that has had H5N1, which seems like a sensible thing to do. So we're completely zero risk, which is great. And this is a very common thing that, we've got our borders, and as long as we do the right thing, we're safe. But of course, the trouble is that Canada and Mexico and Brazil all trade poultry with countries that are infected. And these are live chicks coming into those countries. We have a quarantine loophole between Canada and the US, whereby birds can be shipped in without any quarantine at all, as long as they go to a specific farm in the US. And biosecurity isn't very tight on these farms. They have large fans. Poultry houses have to have large fans to keep the temperature down, and they blast out all sorts of detritus, including infectious material. That's been shown. So this is a false sense of security, and the risk here is 250 to 2,500 infectious bird days a year. Now, when you look at migratory birds, and what the US government had done at the time was to say, they held a little meeting of the heads of some agencies and said, okay, the virus is in China, it's in Europe. How's it going to get into the US? Well, common sense tells you birds migrate from Asia through Siberian migratory pathway into the US. So that's how it's going to get here. So the US government put a lot of money, and you probably remember seeing this in the news of lots of students going out to Alaska for the summer, catching wild birds, doing cloacal smears, looking for H5N1 avian influenza. When you do the analysis, the number of birds that actually migrate across there at the right time of year from countries that have had the virus is about 3,000, and that gives you a risk of 2 bird days per year. So it's again 2 to 3 orders of magnitude lower, and yet that's where we're putting all our money. Now I thought that was a really good result, and we published it in PNAS, and the day after it was published, we got a phone call from the Government Accountability Office, and I thought, uh-oh, this isn't good. Well, it was good. They wanted our data, they used it to analyse how USDA actually do surveillance for poultry. So I think maybe they've used it to some good. Now that's for the sort of international spread, multinational spread. That's the easy pathway. Now let's get into some of the harder stuff to do. The local scale emergence. Now let's just look at West Nile for a minute. This is how complex it gets. Now one of the things about West Nile is it rapidly moved across the US. But each year you get different foci of outbreaks. And you remember 2003 was a huge year in the Midwest, or further west, for West Nile virus with 12,000 horse cases and over 2,000 people. Now how do you predict, using this sort of strategy of trying to predict things about emerging diseases, how do you predict how significant a disease is going to be in a region next year? Very difficult. And if you look at West Nile, it's incredibly complex. You've got over 100 bird species, each with a different level of competence. You've got a numerous mosquito species, some specialising in birds, some specialising in feeding on mammals, some that bridge between the two. And then you've got all these factors that affect mosquito abundance and therefore the capacity to spread West Nile in a region. But what we did is we looked at all of the factors and we tried to boil them down into a few simple measurements we could take in the field and start to study it. So we don't need to measure rainfall because rainfall affects larval habitat for mosquitoes and produces more mosquitoes. So as long as we count mosquitoes in a region, we're safe. We need to know what mosquitoes feed on, which we're in a bit of a black box about. For birds, we need to know how competent they are and how much virus they produce in the bloodstream, as well as some of these other factors. All the ones that are crisscrossed, they're the ones we need to measure. So we set out to measure those things. Now, we already had a good handle on how competent birds are at secreting West Nile. These are some experimental infections. You need a reservoir competency of more than 1 to be really significant there. And some of these just aren't significant at all. Chickens, for instance, don't produce much virus in the bloodstream. So they don't really fulfill a role of changing the dynamics of West Nile, of increasing the dynamics of West Nile. American crows, which are the ones that die in huge numbers, are extremely competent. But of course, they don't live very long. They die, so they then become unavailable to mosquitoes. So how important are they at actually producing a transmission dynamics of the virus in a region? How do you find out what mosquitoes feed on? Well, we found out there's a really cool technique where you just get mosquitoes with blood, and there's what's known as a blooded mosquito, and then you sequence the blood meal, and of course then you can find out what it fed on. And we do that routinely now. It's a huge amount of work, and one of the interns' roles is to sift through piles of mosquitoes every night and find the ones with blood in and send them off to the lab. And you end up with chipmunks, humans, horses, all this sort of stuff. We set up a series of sites across an urban to rural gradient, and this is Smithsonian Ecological Research Centre, which is pristine forest, large parks in suburbia, more urban settings, and then right in the centre of Washington, D.C. And I thought it was really cool. We were going to do New York, but in the end we did D.C., and I thought, well, this is a really good way to get funding. We're actually right there every day. The Congress folks will see us catching birds. We may even get the DNA from the mosquito that just fed on G.W. Bush as he was jogging that morning. I don't know if we ever did that. We didn't sequence it. And also we've got this incredible diversity gradient. So as you go into the urban centres, you end up with a bunch of European birds that maybe are good reservoirs. Maybe that's one of the things that drives this. It's pretty well shown that in the urban centres you've got more chance of being a case of West Nile, even if you correct for population density. But of course the other thing you've got in urban centres is really good habitat for mosquitoes. So we set about measuring this, and it's 5 years of work. This is just one season, and this is a Julian calendar, so the bold line is the spike in mosquito prevalence during the summer. And these are our urban sites. You can see strong presence of West Nile virus all across the season. And the bar indicates the average 1 per 1,000 infection rate in different suburban sites. But what's interesting is some of these pristine sites where we've been working for 5 years, we've never seen West Nile in any of the mosquitoes. Now the reason for that isn't that somehow the diversity of birds protects us, like this dilution effect that's talked about a lot. We've looked into it. It seems that it's just mosquito abundance. The wrong type of mosquitoes are there for West Nile, and there's not enough to keep the virus ticking over in those populations. So you should live in a forest if you don't want to get West Nile. You'll get a lot of mosquito bites, but you won't get infected. By doing all this work, we were able to really start to get a handle on what drives West Nile virus transmission. This is them all, and what we find is to just look at bird abundance. House sparrows are the most common species across these urban to suburban sites. And look at this American robin abundance, pretty low. Less than 5-10% in each of those sites. But what turns out to be, when you do the analysis of the blood meals, mosquitoes really like American robins for some reason. And they preferentially feed on them to a huge amount, sometimes 30 to 1 over other species. And then if you add into that the fact that American robins are pretty good at producing virus when they get infected, they become the most significant host for West Nile virus in the northeast. So here's crows, absolutely irrelevant for the transmission of West Nile virus. They get infected, they produce a lot of virus, but they die so quickly, probably, and they're not preferred food of mosquitoes. So they don't really do a lot to keep the cycle ticking over. The worst thing you can have in your garden is a bunch of American robins driving the transmission of West Nile and waiting for them to spill over to people. So that was interesting. Actually, that's quite good as well. This is an old slide. It says, in review at science. Didn't get in there, so thought you should know that. Now, every year in late summer, you see these cases of West Nile in the northeast. People in New York start getting infected, and then the city government comes in and sprays all the streets. And that's usually late August, September, October even. Why is it so late in the season? And no one really understood that. So when we plug all of these data into a mathematical model of disease dynamics, of West Nile dynamics, among the different hosts, you start to see something interesting. You see that, first of all, the probability of humans being fed on by mosquitoes increases towards the end of the summer. Which seems really odd. Why would mosquitoes go from feeding on American robins to feeding on people? Sure, there's some other bird out there that they prefer as well. Well, when you look at it, it really, we thought at first maybe American robins disappear and there's just no birds around at that time. Yeah, birds are migrating away. But when you look at total avian abundance, it actually goes up towards August, September. So they've got other hosts. If you look at robin abundance here, it drops off dramatically. Robins start to congregate, then fly off, and they're unavailable to mosquitoes. And it just happens that the next preferred host after American robins for the mosquitoes that transmit West Nile is us, mammals, humans. We're the most abundant mammal around there, the most available when we sat out on our rocking chair at night smoking a pipe. So that's why we get these late summer onsets of West Nile virus outbreaks. But knowing all this information means you can do more than that. You can actually predict. In one of our sites, you can predict, knowing early in the season the number of mosquitoes and the types of birds you've got there, the number of cases and the temporal trend in those cases later on in the year. So we did this, and we did this for Washington, D.C., because I thought, again, this is a great tool for raising money, because we can say to George Bush, here's your risk, you shouldn't go out on your bike between June the 23rd and August the 22nd. So we plugged in the model, and then we got this nice series of cases. This is the risk for humans. And then we went out that year and got data on the cases. So is our model true? Well, actually, really fantastically true. I was really pleased with this. I mean, it's not many cases, it's a handful, but the timing of the rise is almost perfect, and then it drops off. And I remember saying to Mum, well, yeah, it's not bad, but look, we've missed it by a week. What's going on? And of course, that week is, you get infected, you think you've got a cold, you go to your doctor, and he eventually works out over a week that you've got West Nile, and then you become a nice dot on our graph. So I think this is a good example of how you can predict local scale disease outbreaks and emergence. But it's actually incredibly difficult because, as you can see, we've done 5 years of work to do this. What about predicting the emergence of a new pathogen on this sort of small scale? If we can predict local scale outbreaks, can we predict spillover of a pathogen or these little spikes of outbreaks? Well, we started to do that as well, and this took, again, a few years to do, but I want to show you an example of that. And this is Nipah virus. It's a virus carried by fruit bats in Malaysia. Now, it's one of about a dozen or so viruses that have emerged from bats recently, so there's some phenomenon with bats and viruses we don't really understand yet. But the idea was to study this and say, why did this virus emerge? It emerged in Malaysia in 1999, killed about 104 people. It's about 40% of the people that got infected died, so it's a pretty lethal virus. It's like Ebola, it's one of these category 4 biosecurity pathogens. There's no drug and no vaccine, so it's a nasty virus. And if that virus went global, it would be a significant burden to us. So why did it emerge? That's our first question. If we understand why, can we predict anything about its future emergence? So to understand why it emerged, you need to go to the first case, the index case, which is on a farm, a pig farm in the forest of Peninsular Malaysia. Nipah virus went from bats into pigs and then into people. And again, in people, it was a dead-end host. So we were thinking, well, there's two possible ideas here. One is that something's changed about bats. Maybe they've altered their migration patterns and come into a new place for the first time and brought this virus with them. Then the other possibility is something's changed in the way people farm pigs over there. Maybe they've got so dense that a virus in bats can easily get into pigs. Either way, it's a pretty weird scenario. And I was fascinated by this idea that, we know about HIV being a chimpanzee virus. Here we've got a fruit bat virus that's got the potential to get into other countries and cause problems. So the first hypothesis was related to El Nino. And what happens every year in Sumatra, people burn forest, clear land for agriculture, and you get this huge haze event. You can see this from a satellite picture. Here's where the index case of Nipah virus was in Malaysia. And here's Sumatra, and each of these dots is a fire. And this is a NASA satellite picture. And when you land in Kuala Lumpur in Malaysia at the right time of year, you can smell the haze even before you get off the plane. It's really bad. Fruit bats are very sensitive to haze. They fly away from it. So the idea is that they flew here for the first time, bringing the virus with them and leading to the outbreak. Now this was pretty promising because when you look at the timing of the outbreak, late 97, early 98, there's this huge spike in particulate matter. It was the middle of that big El Nino in the mid-90s, and the forest fires were raging really bad. It was one of the worst hazes they'd seen for many decades. And we thought this was a great hypothesis. But the problem was when you look at the index case, actually, no, it's not bad. It's a good correlation with this spike, but it's a little bit early. Anyway, we went out to test this. Now, what you've got to do to test this is catch bats and bleed them and test them for antibodies and look at how they migrate over space and time, see if they change the migration patterns in relation to haze, to El Nino. Of course, catching bats is quite hard. And, I can probably do this a little bit here, but, it gets a bit dodgy when you've got to go right up to, that is not me up at the top of the tree there. But what we had to do was put bat nets across here, catch each bat individually. Now these bats wingspans are like this, so they're big, heavy animals. Then you have to winch each one down, put it in a bag, and then when it gets light, so you do this at night, when it gets light, you can start to test it for virus, then you release it. It's a lethal agent, so you have to wear good biosecurity. Obviously, he's a little bit paranoid there, but he was up in the tree as well at the same time. You have to be careful about things like the tide coming in if you're working in a mangrove swamp, so it can be quite difficult. But it's also exciting. I think that's one of the things that's really nice about this field of infectious disease ecology is that it's cool stuff. You get to handle exciting animals that carry a nasty virus in a horrific jungle. And in fact, I think the worst of all was Raina Plaret, who lives just up the road in Bozeman, worked on Hendra virus in Australia, and we found that the bats there tend to live above water bodies, billabongs. So the only way to catch them, because they fly out, I don't know if I've got a picture of it, let me see. Maybe I don't, anyway, I'll go back. But the only way to catch them was to have a net on a boat out in the middle of the billabong, so you could catch them as they fly out. And only one person could stand up at a time, these little rickety tin boats. Anyway, we did this for a few days and everything was going good. I didn't get on the boat, I'd have toppled it over. But then a couple of days later, some of the rangers called us up and said, hey, come look at this, mate. And we got there, there's a 15-foot saltwater croc that had been living in the billabong as they're balancing on the boat, catching a virus with a bat with big teeth and a nasty virus. So it is sometimes dodgy work. Anyway, what we wanted to do was to test every colony of bats in Malaysia, here with the asterisk, and look at the prevalence of virus in those colonies. And we found that each one had high seroprevalence, suggesting it's a normal part of their fauna, if you like. It's a virus that's been with them for a long time. We've isolated virus from those bats, and we've seen repeated transmission of viruses with no effect on the colonies, and there are no clinical signs of illness when these bats get infected. It's a natural virus. It's like herpes virus for us. It just happens to be really lethal in us. So the next thing was, you know, if this El Nino theory is right, then they can only migrate across from one country to another during an El Nino. If you put a satellite collar on one of them and watch it migrate, and you get the signal back on your laptop, it shouldn't move from one country to another unless there's something forcing it, or the theory is wrong. So we did this. This was the first bat we caught. His name was Colossus, named after the X-Men. For some reason, I didn't do that, but... and he's a cute bat. I really like. These are really nice animals. So we caught Colossus here, and we wanted to see, is he going to fly across? Well, as you can see, sure enough, the first thing he did was fly across. And then 2 bats here at this colony, cross the border, meet again for a glass of mango juice, they eat fruit as the sun sets. So these bats, basically, when you do the home range analysis, they range between these 2 areas on a weekly basis, and they follow the fruiting of tropical fruits as they move along. So that puts another nail in the coffin of that hypothesis. And finally, we found that there's some new cases. The first cases of Nipah virus were actually before the El Nino event. So it looks like this bat idea is wrong. So it's something about the pigs. So the issue there is, how do you go back in time and look at an outbreak in a pig farm when that pig farm's completely demolished? So you can't recreate the outbreak in a lab. This is 30,000 pigs. So the only way to recreate the outbreak is to use mathematical modelling. So we started working with Andy Dobson and Juliet Pulliam at Princeton, who specialise in this. And I think this is really cool stuff. So what you do is you create as some of you in the audience know, you create a computer pig farm, and you measure the dynamics of the virus in the lab in pigs, and you get data on how often pigs give birth, how long they live, etc. And you plug that all into this mathematical model, and then you simulate an outbreak. And we got really good data because these farms were pretty sophisticated in Malaysia. They were designed for export. They had a special license. They had to keep really good data on everything. So we studied a lot of farms. This is the index farm with the first case. We knew where every pig was on that farm, what the birth and death rate, etc. And what we're trying to understand was, if you look at the dynamics of this virus, the virus emerged in January 97, and then for 2 years, it ticked over in the pig farm before it caused this huge outbreak in pigs and people. Now, these are human cases, and because there's no human-to-human transmission, this means that the virus must have been present in the pig farms for 2 years. Now, for a virus like this that's really virulent, it doesn't make sense, because if you introduce that into a pig farm that's high density, they're all going to get infected very quickly. Some of them are going to die. Most of them are going to become immune, and the virus runs out of places to go, and it dies out. And in fact, when we started the model, and this gives you an idea of the amount of data you need to collect to have a really good model. And this is a sort of overview of the model. So each barn producing different age classes of pigs, we had to get all the data for those. So it essentially was a huge series of differential equations to describe the dynamics of the virus. Now, when we ran the infection on the computer, when you introduce the virus on a single introduction, if you just look at the green here, that's the infectious pigs, so that's the number infected over time. You get a peak and then it dies out. And there was no way, whatever we did within the bounds of how pigs are managed in Malaysia, we couldn't get this virus to persist longer than a few weeks. And then we found that there was a paper published that suggested, when you look at the phylogeny of the virus from the beginning of the outbreak to the end of the outbreak, there's actually a few changes, subtle changes to the sequence, to the RNA sequence in the virus. And what the people who published this paper suggested is that the virus has evolved over time. And we thought, well, yeah, it could have evolved, but maybe these bats are repeatedly visiting these farms, and maybe they're reintroducing the virus a bit later, so it's a different strain of virus. So we put that into the model, and when you introduce the virus for the second time, you get this weird effect. You get a much lower spike, and then the virus persists for a very long time, just like we saw in the outbreak. So why is that? Well, what we think's happened here is that in the initial outbreak, you get infected female pigs that produce piglets. And those piglets carry maternal antibodies. Now, some of those infected female pigs die and then are replaced by new ones as the farm continues to produce. But these piglets with maternal antibodies are still present. When the virus is introduced again, it's now introducing it into a partially immune population. And it's not only that, it's a population where the piglets start to lose the maternal antibodies, become susceptible, and then allow the virus to continue ticking over. And you can sort of do an analogy, it's a bit like a fire. Your initial fire is very hard burning and then dies out. But you've got a few wet logs on there, and these are the piglets with the antibodies, and they start to dry out. You put another spark on the fire, and gradually it builds up again, and as logs dry out, the fire ticks over. So this epidemic, instead of raging and then dying out, started to smoulder and persist for 2 years. And that's why we think the virus emerged. So what we saw there is emergence due to agricultural intensification, another type of change that we're doing all around the planet. And it's actually something that's been proposed for H5N1 in poultry farms. If you vaccinate a population, don't vaccinate them completely, you may produce this sort of dynamics that can lead to a much longer circulation of H5N1. Now, what does this mean for predicting? Now, this is what, if you look at pig production over time in Malaysia from 1960 to now, you see this nice rise, it's becoming wealthier and wealthier. Then this red line is when Nipah virus emerged, and the government culled out all the big pig farms, so they lost a lot of production. But if you look at mango production drops off at the same time. Now, the reason for this is in Southeast Asia, when you produce pigs, you use the manure to fertilize fruit trees, and you plant them around the pig farm, and you get an extra profit from your crop. But of course, those mango trees bring in fruit bats, feeding on them at night, and they spit bits out. They're notoriously messy eaters, and spit bits of pulp out into the pig farm. So that's how it seemed to emerge. This dual system of mango and pig production, and a very certain intensive type of pig production. But of course, the government of Malaysia wants to start these pig farms up again, and they talked to us about, well, could we do this without a risk of Nipah virus? So we looked into it, and actually it's possible to work this out. We know that transmission is from bats to pigs. We know where all the bat colonies are. We know where all the pig farms are. So some are lower risk. There are no bats there. We know that only certain types of fruit trees attract bats and were associated with the outbreak. So we got data on fruit production over Malaysia, and then we know where the risk is in terms of fruit production. And then we know the size of the farm, because it's the very big intensive farms that are the high risk for the virus first spilling over. But it's these smaller farms in the south of Malaysia where there's more people per pig that have a higher risk of people getting infected. So this is where the high risk for emergence is, where bats, pigs, and very intensively farmed pigs exist. But then once it emerges, this is where the big risk for numbers of people dying is. So we produce a risk assessment for the Malaysian government now, and you can see pretty clearly the risk of emergence is high here, but the number of cases is going to be much lower than in the south. So you can have the intensive pig farms here, but not here. So simple analysis, a lot of data, and you can really start to prevent the future emergence of a disease. Now, for the last few minutes, I want to talk about my vision for the future. And this is much more dubious science at this point, so please feel free to criticize it openly afterwards. But we're beginning to get to the bottom part of that structure where we're now trying to predict not the local spread of cases or the re-emergence of a disease, but the first case of a new pathogen. And what we're talking about here is, could we have predicted HIV before it emerged and gone out there, tried to block bushmeat hunting or do surveillance in people to stop them and look for these new viruses and prevent it becoming a pandemic? Now, I think that's something you can do. I think one day we'll be able to predict pretty reliably disease emergence, the first new case of a new virus. And it's also a challenge because a very well-known virologist, Fred Murphy, says you can't do it. So that's a nice challenge. In general, there's no way to predict when or where the next new important pathogen might be. Now, I'd been saying this for a while, and then I read the paper. Oh well, maybe you can't. And he does have a point. If you look at these new viruses from wildlife, let's just focus on vertebrates, because most pathogens that emerge into people from wildlife come from birds and mammals, essentially. There's about 50,000 known vertebrate species. If you assume there's 20 new viruses, endemic viruses that we don't know about, per vertebrate, that's 1 million viruses. And just in bats, this group of new viruses in bats, there's 20,000 by this underestimate, almost certainly, alone. Now, we know that there are only 2,000 or so viruses, so simple mathematics, we're underestimating emergence by 99.8%. Now, I just made up the numbers, but just as a way of illustrating it, there's a huge potential for future disease emergence. And how do we really measure on a global scale where these unknown viruses are? That's impossible. Now, our approach was to try and do this by looking at what we know. And what we did is we used a database, at least a list of pathogens that was published. It's every human pathogen. We pulled out all the emerging pathogens, there's about 450 of them, and then we spent a couple of years looking through the literature, finding the first case, or the first cluster of cases for each of those pathogens, where we could. And then if you plot them on a map, you start to see this little cluster of new diseases emerging. So here's trench fever, re-emerged in Mexico City in 1960. California flea typhus in Nueces County, Texas in 1991. If you plot that out globally, this is what you get. So there's 450 dots here. And what you see is really interesting. And I was surprised at this. Where I'm from, which is England, is the dirtiest place on the planet. Well, we knew that, That's why I moved to New York, which is also, unfortunately, not a great place. Now, one of these dots is West Nile. One of these dots is mad cow disease. That's what the database says. But of course, this is totally biased, because what it really shows you is where people are looking for pathogens. It's hospitals, it's researchers looking at infectious diseases. Probably there's been a whole host of clusters of cases of new pathogens in small groups of people that were never identified in the Amazon, in remote parts of Ethiopia or wherever. So how do you correct for that? Well, what we did is we looked at Journal of Infectious Disease, and we went back to 1940 and looked at every single author to the current date and plotted the origin out in our database. So 17,000 authors. So it's a lot of work. But what we could do with that is test out some really important theories. So first of all, are emerging diseases a real worrying trend, or is it just hype? Well, if you look at the raw data, decade by decade, you see a rise. And there's the 1980s, you see an extra special large rise because HIV went pandemic and itself caused the emergence of a bunch of new diseases. But if you correct for bias, you still get a very significant growth over time. So it is a phenomenon, it is a real issue. And what's really interesting is this yellow group, they're the ones that come from wildlife. And if you look at the last decade, they're significantly growing over time, and in the last decade represent the largest number, the largest proportion in the database. 61.4% of emerging diseases come from wildlife. So we wanted to test out some ideas very briefly. First of all, is it really a human-related issue? Is it really things that we do? So if that's true, then richer countries should have more EID events. Socioeconomic drivers should drive the risk. The second thing we wanted to look at was if zoonoses from wildlife are a big threat, then what we should see here is there should be more risk of a disease emerging in a place where there's more unknown pathogens. And here we have to get to a measure of unknown pathogens. Now, if we go back to our idea that vertebrate species carry a certain number of unknown viruses, then all we've got to do is get a database on global, say, mammal distribution. In fact, there was one published a couple of years ago. So we used those data and we tested out the theory. And what we did is we looked at, this is the Journal of Infectious Disease correction factor, this is human population density, population growth, latitude, rainfall, and wildlife species richness. And we split the database out into zoonoses from wildlife, drug-resistant microbes, and vector-borne diseases. Each one of those groups is strongly correlated with human population density. That means the risk of spillover or first case emergence is higher where people are most densely situated. Now that's two things, that's density itself, but it's also the activities that they do in those areas. So it really is an anthropogenic issue. And then secondly, if you look at wildlife diseases, wildlife zoonoses, very strong correlation with only that group to wildlife species richness. Now because this is spatial and because we've got a pretty solid correlation there, we can plot this out on a map. And what you see is, what we're expecting to see is, here's England with very little wildlife diversity. That should not be the most bright hotspot on the map now. These are the areas of global mammal diversity. So we should start to see Africa, parts of Latin America and Southeast Asia become hotter as disease hotspots. And when you do this, when you plot it out, that's exactly what you see. This is the relative risk of a new emerging zoonotic disease from wildlife. And yes, Europe and North America are still hotspots because I suspect because we've done so much environmental change and we continue to do that, we still get things like raccoon rabies emerging and other diseases. But now we start to see really bright hotspots in Southeast Asia, Africa, Latin America. So this is, it's a very preliminary attempt to identify the place where the next HIV will emerge. And in fact, when you look at a distribution of where the people working on infectious diseases are from our data, you can see that the richer countries really do have the biggest effort of surveillance. But we focus a lot on our own backyards. So we're really missing the point. Here's where the diseases are going to come from. And we're not doing enough work there. We're kind of missing the point. I think we need to reallocate the global effort for disease surveillance to some extent. If you just look at those bat viruses, for instance, we know the distribution of those bats. Why aren't we out there looking for new pathogens in bats? Looking for the next one that could become more severe? And in fact, Nipah virus has now emerged in Bangladesh, which is a very bright hotspot on our map. And it's now emerged repeatedly, over 8 outbreaks. The case fatality rate is now over 70%. And worst of all, it's gone human to human. It started this process of becoming pandemic. This is the first case, the index case, of an outbreak 2 years ago in Bangladesh. He infected one of his family members, 3 of his family members, who then infected a relative, who then infected a religious healer. And people, as that guy was dying, people came around to visit him and then set off a large outbreak. So now we've got a disease that's got a fatality rate of 40% to 70% that's beginning to get into this global network, human-to-human transmission. So we're seeing a serious threat. And our current approach now, our plan for the future, is to use some of the new technology like 454 sequencing, where you can pick up not only pathogens that you know about, but by doing blast searches, related pathogens that are unknown right now, and really get to measure the diversity of potentially zoonotic pathogens in those hotspots. And we've already done this with some bats in Bangladesh and found a new virus by just looking at 8 bats, which to me was incredible. We targeted them very specially. We went after pregnant females. The idea they're immunosuppressed, but with just 8 samples found a new virus that belongs to a zoonotic family. And this is our global network of labs that we're working with to start this process of discovering the next important pathogen. So I want to finish there with that hopefully more positive note, and thank all the collaborators, including Juliet Pulliam, who did the modelling with NEPA, and Marm Kilpatrick, who runs the West Nile virus programme, John Epstein, who saw up to his neck in water before, Andy Dobson in India, dressed in traditional garb, for those of you who know him. And also to thank the funders, and to mention our journal, EcoHealth, where we're trying to bridge the gap between ecology and health. And finally, to thank the wildlife, because they put up with a lot from us. At that point, I'm open to questions.
Anyone have any questions for Peter?
Well, I'm not going to go until you ask some questions. So that's pretty sad.
So I have a question. Yes. Well, I'm turning the lights up. So what role does increased surveillance have in apparent increasing incidence? Is it really an increase?
I think we got at it. I think we were able with the. I mean, it's a very crude measure, but just by looking at that growth in the number of scientists over time in the articles in Journal of Infectious Disease, I mean, it's a huge growth, and we corrected for that, and it's still a significant increase. So this really is a phenomenon, and it really is an issue. You know, it's and it's not just a product of us worrying about it and going out there and looking for them.
So, considering the growth in population in the world, how realistic is it that we're going to be able to actually stop emerging diseases or prevent them?
Yeah, well, it's totally unrealistic is the answer to that, which is, that's the attraction of it, you know. I mean, you know, we're working in China now because we found SARS in bats in China. We found that the origin of SARS is probably bats, and they're in the wildlife markets there. So we're working with people in the wildlife markets and testing them and looking for new pathogens and looking at wildlife there. Now, that's 2,000 years, 4,000 years of culture to eat this diverse array of wildlife. It's a huge risk for emerging disease, really hard to stop. But, you know, in the future of somewhere like China is going to be, they're going to become more westernized. People are going to probably eat less of that. There may be a bit of a rise at the moment as people are getting richer and can afford that stuff better, but I think it's going to go down. So ultimately, it's like conservation. Once you go through the horrific deforestation event and industrialization, you know, we now have more forest in the northeast than there was 50 years ago. So I think we're going to go through that sort of cycle. So what we've got to do now is just get ready for it. You know, look at those, increased surveillance in those hotspot areas. It's a bit like an earthquake zone. If you're building in an earthquake zone, you put some protection down. So the protection is have a surveillance program. When a virus spills over, catch it really early, or go out into wildlife and predict which one's going to emerge next and stop people eating them. Work with them and talk about the risk. And we're doing that in Bangladesh now with some of those bat hunters.
So do you think that it's more effective to try to prevent them than it is to try to treat them?
Yeah, I think it is. I mean, if you look at HIV, for instance, I mean, okay, that's an extreme example, but where's the HIV vaccine? You know, we were promised an HIV vaccine 20 years ago. My wife worked on an HIV vaccine for a PhD, and of course it evolved so rapidly, it's pretty unrealistic. We've got HIV drugs, but you know, after 40 million people get infected, and they're still not perfect. Where's the SARS vaccine? We're pumping billions of dollars into SARS molecular biology. SARS could emerge again, and we still don't have a protection. So relative to those billions of dollars, just a little bit of effort focused on surveillance ahead of time. That's the message.
This may be unrelated, but maybe not. You're talking about biology and emerging epidemics. There's also chemistry involved in human-generated toxins. And I was trying to think, you know, this surveillance might be useful for looking at hotspots, strange diseases that emerge. Is there any evidence that these two spheres, the biology and the chemistry, could overlap, that you know, might have a higher mutation rate? It's kind of a wild idea.
No, it's very interesting. I was over in the University of Pittsburgh a few days ago talking to some people who work in Bangladesh on arsenic. Now, one of the things we see in the patients in Bangladesh with Nipah is that they get more respiratory cases than we saw in Malaysia. Apparently arsenic, one of the symptoms is respiratory symptoms. So maybe they're kind of a bit more predisposed to a respiratory infection, and that's probably the way that it's gone to this more human-to-human transmission. So I think there may be something in that side of toxicology. Some groups of people are maybe more susceptible. You think about co-infections with other pathogens too in some of these places. But I've not thought about the other one, and it's an interesting idea. We were talking earlier on, there is so much to be studied on the interactions between stress and disease and toxins and disease. It's a field that's waiting to be captured, yeah.
Could you talk a little bit about the bushmeat trade in Africa? As I understand it, it's really gone through the roof in the last 10 years, but it's not only for local consumption, there's a lot of export.
Yeah.
To Europe and North America.
Yeah, there is. And when the fruit bat diseases started appearing, there was a lot of concern that you can buy fruit bats apparently in London. They're imported in there from Africa. There's a lot of illegal bushmeat brought over. What tends to happen is it's smoked. It's this. It's not really cooked, but it's smoked and then it's dried. So for a virus like that, it's not going to persist in those tissues. The big risk with bushmeat is the people who catch the animal, the people who butcher the animal. And I think that's an issue, because as you get more efficient transport in and out of the forest, and more and more logging roads, more and more hunters, they're getting fresher meat back into the villages quicker, and then it just increases the number of people who are exposed. So we've actually been working with some hunters in Malaysia who used to hunt bats, and we've got them introducing us to the restaurants that get these wildlife and butcher them. We've actually got a program now to look at people who work in those restaurants and see if they're picking up infections. But it's definitely a growing trend right now. Long term, it should go down, hopefully.
When you're trying to get changes done, what's been your most easily openable door? Has it been through governments? Has it been through trade associations or industry groups? Has it been through medical or social outreach?
Well, yeah, the truth is that going through governments is usually less effective. And I think that's one of the problems with these diseases. They become such an issue because they cost such a lot of money economically. SARS, for instance. China is very sensitive about SARS. It's a loss of face to the nation that this disease emerged there and costs so much in lives and economy. So if we were CDC or the World Health, we can't just go into the country and start working in the wildlife markets. But because we're a small group and we do this in a culturally sensitive way, working with local people and understanding the culture and sharing expertise and specimens, we've been better able to do it. And to actually change people's habits, we've had limited success so far, but we've only just started. In Bangladesh, we've been working with the Darul Disease Centre there, who've developed a simple protection system for the guys who collect date palm sap in trees. This is like maple syrup, and they sell it fresh every morning. Well, the bats come and feed on this at night, and that's been one of the keys to starting off these outbreaks. So just a simple, cheap cover that stops the bats getting in there is pretty effective at blocking transmission. So we're working with some of those date palm gotchas, they're called, to stop it. With one of the diseases that I've worked on for years, an amphibian disease that's spread through trade, we've been able to go the other route and produce data that clearly shows trade drives the disease, and then get the disease registered as notifiable through the World Organization for Animal Health, which lists diseases as notifiable. So, you try different ways, but softly is the best.
Have you done any investigation into the area of why a disease mutates in such a way that it actually jumps a species barrier? I mean, you have this area of biodiversity, and you have this kind of an edge effect of people kind of maybe infringing on this area of biodiversity. Are there certain activities they do that disturb and maybe lead to a virus changing or bacteria changing?
Well, that's a really good point. Now, what we've done here is ecology. It's human behavior, social ecology, social behavior. That's one side of it. The other side is, what does the virus do? And some viruses are much better at moving from one host to another, and they're the ones you've got to watch out for. Now, that to me is the big black box in disease emergence right now. And there's been a huge amount of work on the details of viral molecules and how they're able to infect certain cells. But we still don't really understand why some are able to jump host. One idea is that, for instance, viruses like avian influenza switch segments of genes so they can mutate more rapidly. And negative stranded RNA viruses, they tend to be one of the most common groups that have emerged, and they have a higher mutation rate. So a virus that mutates a lot maybe will mutate a version that can get into people from a bat or from a civet or whatever animal. So they're the ones to look out for. Now, I think there are going to be ways to target those groups. What we're doing with the surveillance now is targeting the animals in the hotspots that have been most commonly shown to be reservoirs for emerging diseases, looking at the people who are highest exposed, and then testing for the pathogens that are most likely to emerge. But it's still an inexact science right now.
So just quickly, given the high mutation rates and the lack of conserved genes across viruses, I'm kind of curious what sorts of approaches do you use to go out looking for something that you don't know is there and you don't know what it is?
Yeah, so one thing is, for instance, with that group of viruses, Nipah virus, you look for antibodies in wildlife, and people have found these in bats in Africa now and in China just recently. And because the antibodies cross-react across the whole group of these new viruses, you not only find Nipah virus, you also find other related viruses. And the same with DNA and RNA sequences. If you go to conserved regions of the genes, then you might not only find the virus that you know about, but also the sister or brother virus that's close to it. Finding something completely unknown is tougher.
All right, well, let's thank
Oh, no, all right, there you go. We'll talk after. Thanks.